name is John Christianes Flores from B.S.I.T 1A. So today we will discuss what is gene therapy and stem cells. For your understanding, in gene therapy and stem cells, genes carry the instructions that control how our bodies grow and function. When genes become damaged or contain errors, diseases can occur. To solve this, scientists develop gene therapy and stem cell therapy, advanced medical methods that can repair or replace the damaged genes and cells in our body. So, let's talk about how gene therapy works and the three main ways gene therapy works. The first main three ways of gene therapy is gene replacement, by replacing or substituting a faulty gene with a healthy one. The second way in gene therapy is gene knockout, by inactivating or turning off a gene that doesn't function properly. And the third way of gene therapy is new gene introduction or add new gene, by adding a new gene to help the body fight a disease. To deliver the new genes, scientists often use harmless viruses called vectors that act as carriers. The importance of gene therapy and stem cells. Stem cells are very important in gene therapy because they can be modified with health genes and then multiplied to replace the damaged tissues. Stem cells have two main abilities: to renew themselves and to become different kinds of specialized cells. This makes them powerful tools and in curing genetic diseases, repairing tissues, and even regenerating organs. So, this is a part of stem cells' main abilities: self-renewal. Understanding for self-renewal. Self-renewal means that stem cells can provide and produce identical copies of themselves over long periods. This process keeps a constant supply of stem cells in the body. Without self-renewal, our bodies would run out of new cells to replace old or damaged ones. Differentiation. Differentiation is the process where a stem cells becomes a specialized cells with a specific function, like a red blood cells that carries oxygen, or a nerve cell that transmits signals. These abilities helps repair tissues and organs that are damaged by disease or injury. So, these two is the type of main ability of stem cells. Then, let's talk about the importance of self-renewal and differentiation. Both self-renewal and differentiation are essential for life. Self-renewal ensures there is always a supply of stem cells available, while differentiation ensures that the body has the specialized cells it needs to function properly. If stem cells could not renew or differentiate, healing and growth would be impossible. Stem cells jargon. The specialized technical vocabulary used in the field of stem cells, biology, and medicine. Scientists use specific terms to describe how flexible stem cells are. For example, totipotent can form all cell types, including both body and embryonic cells. Pluripotent can form almost any cell type except extraembryonic tissues. Multipotent can form several but limited types of cells. Unipotent can form only one type of cell. This term describes how powerful or specialized a stem cell can become. So, these are the examples of stem cell application. Stem cells are used in many medical treatments today. For example, hematopoietic stem cells from bone marrow are used to treat leukemia. In the future, scientists hope to use stem cells to treat heart disease, diabetes, spinal injuries, and even genetic disorders. In conclusion, gene therapy and stem cells are groundbreaking discoveries in medicine. They give hope for curing diseases that were once thought to be untreatable. Through continued research, these technologies may change the future of healthcare and improve many lives. So, let's proceed and talk about the types of stem cells.
Good day everyone, my name is Ruel Calderon from B.S.I.T 1A, presenting embryonic system. Stem cells, those are cells that have the capability of self-renewal and differentiation. Self-renewal is the ability of cells to proliferate without the loss of differentiation potential and without undergoing senescence. A stem cells is a blank cells, pressure cells that can give to resize multiple issue types such as a skin, muscles, or nerve cell. A stem cell is essentially the building block of the human body. Three unique properties of stem cells. Stem cells are capable of dividing of renewing themselves for long period. There are unspecialized and there can give size to specialize the cell types. A stem cells is uncommitted until it's received a signal to develop into a specialized cell. Features of stem cell. Stem cells are very unique cells. Stem cells have an amazing ability to develop into several distinct of cell types in the body. The cells can be used a repair system for the body. Stem cells can theoretically divide without limit a live organism in order to replenish virus types of cells. When a stem cells divides, each new cells that the potential to either remain a stem or cell to become other types of cells without a specialized function. Embryonic stem cells of inner cells mass are pluripotent, that is, they are able to differentiate to generate primitive ectoderms, which ultimately difference during castration into all derivatives of the three primary germs layer. They include each of the more than 220 cell types in the adult body. Pluripotency distinguish embryonic stem cells from adult stem cells found in adults, while embryonic stem cells can generate all cell types of body adult. Stem cells are multiple tent and can produce only a limited number of cell types. This would provide a radical new treatment approach to a wide variety of conditions where age, disease, or trauma has led to tissue damage or dysfunction. Embryonic stem cells of inner cells mass are pluripotent, that is, they are able to differentiate to generate primitive ectoderms, which ultimately difference during castration into all derivatives of the three primary germs layer. They include each of the more than 220 cell types in the adult body. Pluripotency distinguish embryonic stem cells from adult stem cells found in adults, while embryonic stem cells can generate all cell types of body adult. Stem cells are multiple tent and can produce only a limited number of cell types. This would provide a radical new treatment approach to a wide variety of conditions where age, disease, or trauma has led to tissue damage or dysfunction. How to drive an embryonic stem cell line. A stem cell line is composed of population of cells that can replace themselves for long, provide us into other body. Isolating ES cells, sperm and egg joint. 2. Embryo develop for 5 to 7 days. 3. Remove inner cells mass. 4. Grow in dish. 5. Change culture conditions to stimulate cells to differentiation into a variety of cell stems. Advantage of embryonic stem cell. 1. Flexible. Approach to have the potential to make any cell. 2. Immortal. One embryonic stem cells line can potentially provide an endless supply of cells with defined characteristic. 3. Availability. Embryos from into vitro fertilization clean. Disadvantage of embryonic stem cells. Difficult to differentiate uniformly and homogeneously into a target tissue. 2. Immunogenic. Embryonic stem cells from a random embryo donor likely to be rejected after transplantation. Tumorogenic. Capable of from tumor or promoting tumor formation, destruction of developing human life.
Hi everyone, my name is Janay Bandakomos. Today I'm explain about stem cell, also cell tissue stem cell. They are special cell in our body that help and maintain our tissue. Number 1. Definition. Adult stem cell, also stem cell, are special cell found in definition parts of the body. Their job is to retain and maintain tissue. Number 2. Main point. They can turn into different specialized cell but only in limited way compared to embryonic stem cell. Number 3. Importance. Adult stem cells are important because they help our body heal naturally. They are also used in medicine like bone marrow transplant for leukemia patients. Number 4. Closing. In short, adult stem cells are our body repair system. They're limited compared to embryonic stem cells, but they are very used for healing and treatment. Advantage of adult stem cell. Number 1. Natural repair system. They help repair and maintain tissue in the body. Number 2. Medical use. Use in treatment like bone marrow transplant for blood disease. Number 3. Less ethical issue. Unlike embryonic stem cell, they don't involve destroying embryos, so they're less controversial. Number 4. Lower rate of reject. If take from the patient's own body and the immune system is less likely to reject them.
My name is Harvey Stasio, 19 years old from Bumbunag, San Gabriel, La Union. My topic is induced pluripotent stem cells or iPSCs. Induced pluripotent stem cells iPSCs. Dr. Charles A. Goldthwait Jr. wrote in his article The Promise of Induced Pluripotent Stem Cells that in 2006 there were researchers at Kyoto University in Japan who identified the condition that would permit specialized adult cells can be reprogrammed genetically to become a stem cells like state. These adult cells called induced pluripotent stem cells are reprogrammed to an embryonic stem cells like state by introducing genes important for maintaining the essential properties of embryonic stem cells. Same as embryonic stem cells or iPSCs are pluripotent and can make all the different type of cells in the body. Turning adult cells into induced pluripotent stem cells involved adding particular genes to cells in the body to make them behave like embryonic stem cells. These genes provide these cells instructions about how to behave. This process is analogous to changing the instructions in a computer program to make the computer do a different task. Researchers anticipate that the use of iPSCs can help treat diseases like Parkinson or Alzheimer in the future.
Good day everyone. I am John Emerson Argapos.
The topic is surrogacy and in vitro fertilization or IVF. Introduction. Surrogacy and IVF are two advanced methods of assisted reproductive technology or ART that help individuals or couple conceive a child when natural conception is not possible. They were developed to address infertility, which affect millions of people worldwide due to medical, genetic, or lifestyle factors. IVF or in vitro fertilization focuses on creating an embryo in a laboratory setting. Surrogacy focuses on having another woman carry and give birth to the baby. Both technologies are medical breakthroughs but raise important ethical, legal, emotional, and social questions. In vitro fertilization, in vitro is a Latin term meaning in glass. It refers to fertilization that occurs outside the woman's body, usually in a laboratory dish or test tube. IVF allows doctor to combine a woman's egg and a man's sperm in controlled condition to create an embryo, which is implanted in a woman's uterus. Step-by-step process of IVF. Step 1. Ovarian stimulation. The woman takes fertility hormones like FSH and LH to stimulate the ovaries. This encourages the production of multiple matured eggs instead of just one. Step 2. Egg retrieval. A minor surgical procedure under ultrasound guide removes the eggs from the ovaries using a thin needle. Step 3. Fertilization. The eggs are combined with sperm in a petri dish. In some cases, doctors use ICSI or intracytoplasmic sperm injection, where a single sperm is injected directly into an egg. Step 4. Embryo culture. The fertilized egg, now embryos, are monitored for several days, usually 3 to 5 days. Embryologists observe their growth and choose the healthiest embryos. Step 5. Embryo transfer. One or more embryos are placed into the woman's uterus using a thin tube. If implantation occurs, pregnancy begins just like a natural conception. Step 6. Pregnancy test and follow-up. After about 2 weeks, a blood test confirms whether the embryo successfully implanted. Advantage of IVF: help overcome infertility from blocked fallopian tubes, endometriosis, low sperm count, or unexplained causes. Allows genetic testing (PGT) to screen for hereditary disease before implantation. Fertility preservation. Eggs, sperm, or embryos can be frozen for future use. Option for all families. Same-sex couple and single parent can have biological children using donor limb tests. Disadvantage of IVF. 1. High cost. Each IVF cycle can be expensive, especially in countries without government subsidies. 2. Emotional stress, hormonal change, repeated attempts, and financial burden can be exhausting. Number 3. Health risk, ovarian hyperstimulation syndrome or OHSS, multiple pregnancies or miscarriage. Number 4. Success rate varies. On average, 30 to 40% of women under 35, lower for older women.
What is surrogacy? Surrogacy is a process in which a woman, called the surrogate mother, carries in
Hey everyone, my name is Aldrin F. Carubio and today I will be presenting about umbilical cord stem cells. This topic highlights one of the most remarkable medical discoveries that not only saves lives today but also hold promise for future medical treatments and research. So let's start. What is the umbilical cord stem cells?
The umbilical cord is a tube that connects the baby to the mother during pregnancy. It brings oxygen and nutrients and removes waste. After birth, it is clamped and cut. What are stem cells? Stem cells are special cells that can change into different types of cells, such as blood, immune, or tissue cells. Umbilical cord, blood, and tissue are rich sources of these stem cells. What are the types of stem cells in umbilical cord? Number 1 is hematopoietic stem cells. It form blood and immune cells. Number 2 is the mesenchymal stem cells. It develop into bone, cartilage, muscle, and fat cells. The last is the endothelial progenitor cells. It help repair blood vessels. In the umbilical cord, you can find three main types of stem cells. Hematopoietic stem cells form blood and immune cells. Mesenchymal stem cells can develop into bones, cartilage, muscles, and fat. Endothelial progenitor cells help in repairing blood vessels. This variety makes umbilical cord stem cells very valuable in medicine. The collection process. After the baby is born, the cord is cut. Doctors collect the cord blood in a bag, which is then processed and stored in special banks for future use. How this cells uses in medicine. These stem cells are already used to treat more than 80 diseases, especially blood disorders like leukemia and lymphoma. They can also rebuild the immune system after chemotherapy. Scientists are also studying their use for diabetes, heart disease, and even spinal cord injuries. Advantages: the collection process is safe and painless. They also have a low risk of rejection and transplants compared to other resources. Plus, they can be stored for years. Limitations: however, there are also some limitations. One cord may not provide enough cells for an adult patient. Private storage is costly, and while promising, many treatments are still under research. Conclusion of this study: umbilical cord stem cells are a valuable medical resource. They are already saving lives today and will hold the key to curing more diseases in the future. Thank you.